Hi, today we'll talk about the embryology of the chick embryo and the embryogenesis of a telomere egg. You can see normally how we can color these eggs like in Easter holiday, and sometimes you can see two yolk inside one when you fry egg in one egg. You can see the developmental stages of the chick embryo live in situ on the top of the egg, and as well you can see in the bottom infograph the different developmental sketch of the different developmental stages that occur inside the egg. Here as well you can see how scientists can break a hole inside of onto the shell, and they can watch embryo from this hole to see the developmental stages of the chick embryo in situ. Here you can see the structure of the typical egg of a chick. You can see there is a concentric circles or layers of yellow and white yolk around the structure in the middle called latebra. This yolk is covered by a multiple layer of albumin, two thin layers, one inner and one outer layer of albumin, separating in the middle between the two layers of albumin there is a fibrous layer. These albumin become twisted at the sides of the yolk to form a structure called chelaza. After that, this albumin layer is totally covered by another layer, which is the shell layer for protection. And you can see how the shell layer is separated at some point with the albumin leaving an air space. Looking more similar to the structure of the egg, you can see the yolk and the cytoplasmic part is shifted to the periphery due to the huge amount of yolk, and we term it here blastodisc since it's on the top of the yolk. And you can see the chelaza and the albumin layers, the egg white. If you look closer into the layers that surround the egg, you can see that there is inner shell membrane layer and the outer shell membrane layer, and covering by a more solid layer, which is the shell and the cuticle, and all of this for protection. Here you can see what kind of cleavage occur in birds or chick. Actually, the egg is either called a telolithecal or a megalithecal egg due to the huge amount of yolk it contains. So if we section the yolk material, we can see that the embryonic disc, and this is actually the region where the embryo develop, is located on the top of the yolk. So we call it embryonic disc or the germinal disc. And this part only which is affected by the cleavage due to the bulky amount of yolk that remained uncleaved. Soon after fertilization, cleavage start. The first one is vertical, and the second one as well is vertical, but at right angle to the first one. For the third one, it moves across the second one and parallel to the first cleavage. For the fourth cleavage, it's an irregular circle cutting the blastomeres into 8 central delimited blastomeres and 8 peripheral continuous ones. After the fourth succession of cleavage becomes all of them are irregular. Soon after cleavage, and this shows the major difference between the three types of blastula, the blastula of an animal called Amphioxus, which is egg is isolocystal, very few amount of yolk, and the blastula of the frog, which the type of egg is mesolocystal, shifted to the vegetal hemisphere, but the cleavage occur in the bottom cells, the vegetal cells. You can see the main difference is in the formation of the blastocele, which in the case of Amphioxus, the blastocele is all over the large cavity, while in the frog it is shifted to the side. In case of the chick embryo, you can see since all the cleavage occur in the germinal disc, which is shifted to the side of the yolk, you can see that the blastocele is limited to this region as well on top of the yolk. And now the cells on top of this blastocele are called blastoderm. So now you'll have the egg looking from above is divided into three regions, the area biliosida and area opaca and subgerminal cavity. The area pellucida actually is 1 or 2 cell thickness in center of blastoderm, while the area opaca is a peripheral ring of blastoderm cells that contains cells resting directly on the yolk. While the subgerminal cavity actually it's a fluid-filled space between the blastodisc and yolk. You can see it from this figure, it's a section in the yolk and the top blastoderm layer. To the aim to the formation of a blastocele cavity, which will be circled around with blastomeres, so cells will delaminate, just fall down from the blastoderm to form the hypoplast. So eventually, due to the delamination of the cell, and it will arrange in one layer, so a hypoplast is formed, and eventually 2 layers will be formed, an upper epiplast layer and the lower hypoplast layer. These hypoplasts are formed actually by the delamination from cells of the epiplast layer. Epiplast is an epithelial sheet that will future form the 3-germ layer, and now the blastocele is formed between the epiplast and the hypoplast. You can see in this figure as well that the posterior marginal zone is very evident, from which this posterior marginal zone will project some cells internally and merge with the delaminating cells, and from the cells migrating from the deep cells of the posterior region to form the hypoplast. You can see here as well cells from the entire epiplast converge to the posterior midline. So you can see here, and you can differentiate as well from the top figure, the difference between the area opaca, which means that these cells sits on yolk, and the area pellucida, that means that these cells stay or sits on the subgerminal cavity, because still there is no blastocele have been formed, still there is no hypoplast have been formed. So you can see here the cells all from the epiplast converge and migrate to the posterior midline. This leads to a thickness of the cells that raise a little bit above the surface of the cells, which we call it a primitive streak. It starts to take shape. This primitive streak or this thickening of cells will start to migrate to the anterior, making a thickness of cells that soon will give rise to 2 ridges on the sides and the groove in the middle, which we'll name primitive groove. So subsequently, the cells move anteriorly to form the primitive streak, which is collectively called the primitive streak. The origin of the primitive streaks mark the posterior of the embryo, so it originates from the posterior and migrate to the anterior. The opposite end marks the anterior. The dorsal side is where the cells migrate inward. The groove marks the right and the left, so the primitive groove is actually the right and the left. So what's Hensen node? Hensen nodes, it forms at the anterior tip of the primitive streak, and cells involute and ingress through the primitive groove into the blastocele to form the mesoderm. So actually the primitive grooves in the chick embryo corresponds to the blastoporal lips in the amphibian's embryo. So let's talk about the avian blastulation and avian gastrulation. So in avian blastulation, there is 2 layers, actually upper epiplast and the lower hypoplast form. This hypoplast forms by delamination of cells from the epiplast. The epiplast actually is 3 epithelial sheaths that will form the 3-germ layers. The blastocele actually is the narrow space between both these layers, epiplast and hypoplast. While during gastrulation, cells from the entire epiplast converge to the posterior midline, as we have told before, forming the primitive streak. Subsequently, these cells move anteriorly to form a ridge, the primitive streak. Hensen node forms at the anterior tip of the primitive streak, and cells involute and ingress through the primitive groove into the blastocele to form the mesoderm. The gastrulation is complete in the anterior portion and continues to progress in a posterior movement as the Hensen node regresses. So you can see here the arrows show the movement, and actually you can see a section where you can see that cells, the red cells, have ingressed through the ridges of the primitive streak, the ingressed inward, these migrating cells will form the mesoderm. And you can see that cells of the epiplast and cells of the hypoplast will, is surrounding the blastocele cavity. So the primitive streak arises by migrating of cells of the epiplast that represent epipoly. Cells streams toward the primitive streak and then turn under to spread out as a sheet between epiplast and hypoplast, forming a mesoderm. Some involuting cells displaces hypoplast and form endoderm. And you can see from the figure, some of the yellow cells just displaced the hypoplast and got located between the hypoplast and your future form the endoderm. Again, you can see here, it's another diagrammatic sketch showing the difference between the area pellucida and area obliqua. You can differentiate between them because area obliqua cells are sitting on top of yolk, while area pellucida you have a cavity beneath the area. That's why it's a pellucida, because pellucida means clear. You can see as well in the bottom figure the migration of the cells all over the primitive ridges of the streak, and you can see they can migrate inward. These migrating cells give rise to two types of cells: a mesodermal cells that lodge itself between the epiplast and hypoplast, and the endodermal cells that displaces some of the hypoplast to form endodermal layer. So the primitive streak is characteristic for avian, reptilian, and mammalian gastrulation. These streaks arise from local thickening at of the epiplast at the posterior edge, as we said before, of the area pellucida. And this region is called color sickles, KS, and the epiplast and the epiplast above it. What's the check organizers? You can see from this simple figure the relation between the area obliqua and area pellucida, where is color cells locate, the marginal zone, and in the middle there is of course on top the epiplast, below it is a hypoplast separated by the blastocele. So the organizer of the chick embryo, Henson's node, forms from cells located just anterior to the posterior marginal zone. This KS is a local thickening of cells at the posterior edge of the upper layer of the area pellucida, the epiplast. It induces the formation of primitive streak, PS, and Henson node. So you can see here from the figure, you have an egg with before cleavage. Once there starts the cleavage and finishes, you can see that the red region represented the area pellucida, while the green region represented the area obliqua. You can see that color cells starts at the posterior marginal zone of cells from thickening of the cells. The epiplast and the middle layer in the anterior portion of the KS becomes the Henson node. This posterior portion of KS contribute to the posterior portion of the primitive streak. You can see here from a side figure, you can see if you look at the sidewise, you can see where is the marginal zone located with respect to the area obliqua. And you can see that the posterior marginal zone anterior to the posterior marginal zone, you can see the thickening of cells represented by color sickles, and you can see the relation of the epiplast and hypoplast. Actually, this figure trying to pinpoint where a color sickle exists. The Henson node has long been known to be the avian equivalent of the dorsal blastosporal lip. It is the region whose cells become the pre-chordal plate and chorda mesoderm. It is the region whose cells can both induce and pattern a second embryonic axis when transplanted into other location of the gastra of the chick embryo. It's the same like the experiment which I have done in the previous lecture when we moved the dorsal blastosporal region into another embryo, and it developed into an embryo with a secondary axis and a second neural tube. So actually, this it is stressed here that the Henson node is parallel to the dorsal blastosporal lip. This Henson node, it expresses the same marker at the marker genes as the Spearman organizers in amphibians. Moreover, the Hensen node can induce neural tissue when grafted into fish, amphibia, or mammalian embryo. And you can see here when they transplanted the area of this Hensen node or collar sickle, they transplanted from the posterior marginal zone, they transplanted to another region in another embryo. They found that another primitive streak is induced to form. In another experiment, they transplanted the Hensen node from a duck embryo into a region in the chick embryo. It's not the corresponding region. It's just a region in the epiplast, another region. These transplanted Hensen nodes induce the formation of another neural tube beside the host embryo. So you can induce a new embryo by transplantation of Hensen node. Hensen node from the duck embryo is transplanted to the epiplast of chick embryo. So as you see from the third figure, that a secondary embryo is induced, as evident because there is a neural tube have been formed. Looking more deeply into the formation of the blastoderm, you can see this like a side representation of what is happening, and there is a color coding, and this is from the other one from the ventral side, what you can see, and this is from the dorsal side. So you can see here that it starts with epiplast. You can see that the hypoplasts start as islands because they are delaminating from the epiplast, and you have the posterior marginal zone with the collar sickle, which is the thickening of cells. So hypoplast island can be seen as well as aggregation of hypoplast around around KS. You can see here that hypoplasts start to extend, trying to forming a layer and got connected to the posterior marginal zone. So just prior to primitive streak formation, the hypoplast island have merged to form the primary hypoplast layer, which meets the endoplast cells and the primitive cells at the KS. You can see this hypoplast represented in the on the right figures by the dots in green. Initially it was like island, then start to form from the posterior a sheet of cells that migrate to form a complete sheet at the anterior. Finally, you can see that it got connected with the primary hypoplast cell, the green ones, got connected with the blue ones, which is the secondary hypoplast cells coming from the posterior marginal zone. And then the secondary starts to migrate anteriorly to form a complete layer. After that, it starts the primitive streak in the middle layer, represented by the blue color here, by the red color here, starts by stage 2, 6 to 7 hours after the egg is laid. The primitive streak cells, which we said before is like a local thickening of cells, forms a third layer that lies between the hypoplast and the epiplast. By stage 3, up to 13-hour post-laying, the primitive streak has become a definitive region of the epiplast, with cells migrating through it to become the mesoderm and the endoderm. And you can notice from the figure that yellow cells and red cells start to migrate inward through the primitive streak and got internalized and lodged themselves between other cells. The mesoderm between the epiplast and hypoplast, and the endoderm lodge itself inside the hypoplast cells. So if we look closely into the migration of endodermal and mesodermal cells through the PS, the primitive streak, you can see here they labeled the cells which are migrating, and you can see here the direction of movement starting first from the primitive streak that migrate from the posterior to the anterior, and then cells from this primitive streak, which appears labeled by the yellow dye or yellow fluorescence. You can see that it migrates from exterior to interior, and you can see how, by looking to the arrows, how the migration occur. Again, it's another figure showing the arrows, the movement, which is color-coded, how the cell migrates rolling over the primitive streak. The red cells is the mesodermal, and the yellow cells is the endodermal. In this slide, we summarize the developmental stage of the chick embryo. You can see that the start of thickening at the posterior edge of the area biliocida, which is called the blastoderm, the posterior region of the blastoderm. Soon after that, this thickening start to migrate to the anterior, forming the primitive streak, which is taking shape while migrating to the anterior. Soon after that, the primitive streak reaches the anterior, with the head of the primitive streak is called Hensen node. These three stages show the dorsal view and formation of the formation and the elongation of primitive streak. In figure D, you see that the head process starts to develop with the regression of the Hensen node and the primitive streak. Soon after that is further development of the head region through the formation of the ectoderm of the head fold and the anterior border of the mesoderm and the neural fold and the notochord, and starts to appearing the somite on the sides of the neural tube formation, and the primitive streak is more regressed to the posterior. At the end, you can see that the neural folds start to be more clear, and the somites start to be segmented, and the endodermal has now ingressed beneath the epiplast, and the primitive streak is markedly reduced. And these three figures show the formation of notochord, mesodermal somites, and BS regression, primitive streak regression. If you look again to a diagrammatic figure, you can see that where is the Hensen node located at the beginning of before the formation of the head fold, and soon after the formation of the head fold and the somites start to appear on the sides of the neural tube, the primitive streak start to regress to the posterior with the Hensen node until the somite starts to appear and the closure of the neural tube. So actually, if we talk about the chick fate map, it's very hard to recognize the fate map, contradicting to the amphibians, which we can recognize the fate map even on the egg itself. But since the chick embryo, the blastoderm cannot be mapped, as the early chick embryo is small and there is much more cell proliferating and extensive cell and tissue movement, the early fates are not known. Later, when the primitive streak forms, the three-germ layer can be mapped as seen from the figure. So once the primitive streak formed, you can see that the Hensen node will give presumptively the notochord, will direct the formation presumptively of the notochord and somites. And you can see in the gray region, which will give the extra embryonic mesoderm, and the orange region will give the somites and lateral plate, while of course the dark blue will give the neural ectoderm and the light blue will give the ectoderm. Here you can see the neural tube formation in the chick embryo. It starts, of course, with neural plates. On the side there is a neural crest which is bounded by the epiderms, and soon a groove is formed induced by the presence of the notochord beneath it, and this groove, this folding occur at the medial hinge region. Soon after that the folding starts and the ridge starts with the elevation of the neural crest. Soon after that, these conversions of the neural groove trying to close the neural tube and form a complete neural tube, the conversion of the neural folds occurs as the dorsal lateral hinge points cells become wedge shape, and the epidermal cells push toward the central. Soon after that, the neural crest, which is the ridges of the neural fold, which link the neural folds with, which link the neural blades with the ectoderm. Soon after that, the neural crest cells link the neural tube with the epidermis, as seen from the figure. The closure happens and the neural crest then disperse, leaving the neural tube separate from the epidermis. So what is the neural crest? This slide shows the ectodermal derivatives in the chick embryo. The ectodermal can, from the ectoderm, the neural tube arise, neural crest, and the surface ectoderm or the epidermis. From the neural tube, you can see the brain, neural pituitary, and the spinal cord and motor neurons and retina are differentiated. From the neural crest, a lot of structure as seen is differentiated, including the dentine of the teeth, facial cartilage, melanocytes, adrenal medulla, and the peripheral nervous system, including Schwann cells, neuroglial cells, sympathetic nervous system, and the parasympathetic nervous system. While from the surface ectoderm, the epidermis, a lot of structure appear like the epidermis, hair, nails, sebaceous gland, olfactory epithelium, mouth epithelium, including the anterior pituitary, tooth enamel, and cheek epithelium, and lens and cornea. In this slide, you can see the 24-hour chick embryo, and you can see the cephalic head region has undergone the neurulation, while the caudal region are still undergoing gastrulation. And this is because actually the neurulation starts early in the anterior part with the regression of the posterior groove. So you can see that it is highly developed anteriorly more than posteriorly. And you can see the location of each germ layers, ectoderm, endoderm, and mesoderm. The mesodermal development in frog and chick embryo, actually it's both of them, frog and chick embryo, follow the same pattern. So you can see the chick embryo have the neural tube in the middle covered by the ectoderm. On the sides of the neural tube there is the mesodermal somites, which will give to the lateral mesodermal plate. This all on top of the endoderm, showing the primitive gut. You can see as well the notochord is lying just beneath the neural tube. So hypothetically, if you do these cuts on the sides of this germinal disc and you roll this size in the form of the tube, you will see a very similar structure to the one we have discussed in the frog embryo with all the same pattern. So basically both of them share the same pattern of formation of differentiation of mesoderm.